The Coffea Charrieriana, also known as Charrier coffee, is a species of flowering plant from the Coffea genus. It is a caffeine-free coffee plant endemic to Cameroon in Central Africa. It was found to be the only caffeine-free plant in Central Africa, and the second to be found in Africa overall. Previously discovered in Kenya, the International Institute for Species Exploration at Arizona State University and a committee of taxonomists and scientists has voted the C. Charrieriana as one of the top 10 species described in 2008. C. Charrieriana was discovered in 2008 and the findings were published in a paper named A New Caffeine-Free Coffee from Cameroon to the Botanical Journal of the Linnean Society. The plant was named by authors of the paper, Pete Stofflin and Francois Anthony, in honor of Professor A. Charrier, who had made significant efforts towards the coffee industry. His work included coffee breeding research, and he was Director of Research at National Institute for Agricultural Research, INRA. As a result of collaboration from Institute of Research for Development, IRD, Biodiversity International, Paris Museum of Natural History and the French Agricultural Research Center in 1966 to 1987, cuttings from C. Charrieriana were first collected in 1985 from Bokossi Forest Reserve in Cameroon in Central Africa, along with 70 other Coffea species, though the C. Charrieriana was identified to be morphologically different to previously identified Coffea species. Further work was not done until 1997. In 1997, the cuttings were sent to Institute of Research for Development, IRD, in which further study was undertaken. It wasn't until 2008 that, after morphological and genetic studies of this species, was it recognized as a new species of Coffea. Genotyping analysis reveals C. Charrieriana to have diverged from a common ancestor 11.15 million years ago. The C. Charrieriana can grow up to a range of 5-10 m in height and spreads 5-7 m. The shrubs can grow to 1-1.5 m high. The C. Charrieriana has small and thin leaves that are elliptical in shape. The leaves' petioles are 2 mm long. Its leaf blades are 4 to 8 cm length, x 2.2 to 3.5 cm breadth, in dimensions, and 3 to 7 secondary nerve cells per side of the midvein. Anatomically, the leaf structure consists of an upper epidermis, palisade mesophyll, spongy mesophyll, and lower epidermis, similar to the structure found in other Coffea species. However, comparatively to other Coffea species, the leaves are thin at 100-130 μm thick and contain very few secondary nerves. These properties differ from other Coffea species found in Central Africa. This plant consists of two fruits that each contains one seed. The fruit is 9 to 10 mm x 7 mm in size, whilst the coffee seed inside is elliptic in shape and covered in a seed coat made from parenchymatic. Comparatively to other Coffea species, the C. Charrieriana lacks scleroids in its seed coat. The seed measures 5 mm long x 4 mm wide x 3 mm thick. The flowers have no stalk and consists of 5 petals. In the flower, the anthers and style protrude out. The anthers are also attached to the corolla, characteristic of other Coffea species. It possesses a corolla tube, 1 mm long, style, 10 mm long, two-lobed stigma, 2 mm long, anther, 3 mm long, and anther filament, 2 mm long. Biochemical analysis of the seeds reveals that they are caffeine-free. Compared to other Coffea species, C. Charrieriana has significantly lower linoleic acid percentage, lowest polyunsaturated fatty acid content, less than 30%, and 0.8% dry matter basis. As a result, though originating from Africa, the C. Charrieriana is closer phylogenetically to Madagascan than African species, Dussard et al. 2008, 2953. By examining C. Charrieriana's leaf components, it forms separate genetic clusters to the C. anthonii, C. arabica, C. coniferaa, C. humilis, C. capicata, C. liberica, C. liberica var liberica, and C. mannii. C. Charrieriana also has lower caffeoyl quinic acids, CQA, than other Coffea species. C. Charrieriana is classified under the Rubiaceae family and the genus of Coffea. There are currently 120 species of Coffea, in which 2 species, C. arabica and C. coniferaa, dominate worldwide coffee plant production, making up 99% of produce. This plant is endemic to West Cameroon in the Bokossi Forest Reserve. It grows in a habitat of wet rainforest on rocky slopes of an altitude of 160 m. The Charrieriana cannot be cultivated in cold temperatures, requiring a minimum of 15 degrees Celsius for growth. Best cultivation involves frequent watering, moist soil and full sunshine that is unfiltered from windows. For plantation, the seed should be soaked overnight before being planted in moist soil. For cuttings, a 15 to 20 centimeters tip should be inserted into moist soil. The Charrieriana emits a strong smell and the lack of caffeine makes it more susceptible to pests such as birds, spider mites, and insects. Flowering is expected in spring and summer. After 7 to 8 months from the time of blooming, the seeded fleshy fruit of the plant which contains the coffee beans can be harvested. Similar to other Coffea species, the fleshy fruit of the Charrieriana contains edible beans. These can be prepared by drying, roasting or grinding, generally to make coffee. As a naturally occurring caffeine-free coffee, it provides an alternative over decaffeinated coffee which has undergone chemical processes to remove or lower the caffeine. Another possible use is extracting 5-caffeoylquinic acids, CQ8, from the coffee leaves. As for most Coffea species, including the Charrieriana, there contains natural antioxidant compounds. This natural antioxidant can be used in food and nutraceuticals. Coffea Diversa Farm in Costa Rica is currently cultivating Charrieriana. References